Okay, this next short video is going to talk about different aspects of sex chromosomes and sex-linked disorders or diseases. So first of all, I'm sure most of you are aware that humans have a chromosomal way of determining sex. And we've looked at the number of chromosomes that human cells have. They have 46 chromosomes, 23 pairs of chromosomes. And the last pair, the 23rd pair, is the sex chromosome. So pairs 1 through 22 are the autosomes, and the last pair, the 23rd pair, is called the sex chromosomes, and they exist as either X chromosomes or Y chromosomes. Females, get my pen working here. Females in mammals have 2 X chromosomes. They also have all the other 44 chromosomes, and then they have 2 X's. And males have 44 chromosomes and then an X and a Y. And it's the Y that is responsible for determining maleness in humans. Everybody has an X. You can see females have 2, males have 1, but it's the Y that makes the difference, that makes males males. All right, so then let's look at what happens in the when the sex of a child is determined. So here's a female on the left side. This is a weird Punnett square. They've put it on an angle, but anyway, she's going to make eggs, and each of her eggs will have one chromosome 1, one chromosome 2, one chromosome 3, one chromosome 4, blah, 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 all the way up, one chromosome 7, 8, 9, 10. One chromosome 21, one chromosome 22, and then she will have to pick one of these X chromosomes to go in each of her eggs. So some of her eggs will get this X chromosome and some of her eggs will get that X chromosome, but either way they all get an X. And so that's why what this is showing here is that the female eggs have 22 autosomes plus an X. And the X is shown here as this red chromosome. Not really red in real life. All right, so the same deal with the man. He's going to make sperm, and he's going to make, in every single sperm cell, he'll put one chromosome 1, one chromosome 2, one chromosome 3 to make, a haploid cell, all the way up to 20, 21, 22, and then he gets to put a sex chromosome in the sperm as well. Half of his sperm will get the X, and half of his sperm will get the Y. So that's what this is showing here. Here's an X carrying sperm, and here's a Y carrying sperm. All right, so 50% of the sperm carry X's, 50% carry Y's. Then it's basically a race, and it depends which sperm, this one or this one, fertilize the egg. If this sperm gets to the egg first, there's an X from the mom, and the sperm brings another X. Then you have 2 X's, so the baby will be a female. If this sperm gets to the egg first, then the mom put an X in the egg, the dad brings a Y by way of the sperm, and then you have an XY, and so therefore the child will be a male. And if you look at this, you can see because 50% of the sperm are X's and 50% are Y's, if all other things being equal, if it's just a straight out race, half of the time the X's will win, half of the time the Y's will win, and that explains why overall in the general sex ratio in the population, you have a 50-50 split of 50-50 girls to boys. All right. So that's the actual genetics behind sex determination. It's the sperm or the man that controls whether it's a boy or a girl that is conceived. All right, so let's move on to the next image here. So here we're zooming in on just the X chromosome. All right, the X chromosome is quite long. It's quite a big chromosome. The Y chromosome is very, very, very short, very short. Even though they're supposed to be a pair. They're not like the other chromosomes. The autosomes, the pairs match, but the sex chromosomes, they don't, and that causes some issues. So first of all, any gene that is found on an X or Y chromosome is called sex-linked because it's on the chromosome that is to do with determining sex. There aren't that many genes on the Y because it's so tiny. And actually, you can't have any genes on the Y chromosome that are essential for life because a fair number of the world's population, half, don't have a Y, and they do totally fine. Like, I do not have a Y chromosome, and I'm totally fine. I can survive. So there's nothing on a Y that you have to survive. But there are plenty of things on the X chromosome that are essential to life. You cannot survive without at least one X chromosome. So let's have a look at this. You can see here a list of things that you find on the X chromosome. And I love this slide for a couple of reasons. So look at, these are all names of diseases. Ichthyosis, that's like a skin condition. Ocular albinism, Duchenne muscular dystrophy, retinitis pigmentosa, hemophilia. You know, these are all, some of these are pretty nasty diseases. And if you were to look at this, you might think, oh my gosh, who wants a gene? Who wants genes? Genes just make you sick. All genes are there for to give you diseases. And I think this is a misconception that's out there in the public, is that genes cause disease, right? Now, let's look at the flip side of that. The names of these diseases, or the names of these genes that you see listed here, these aren't really genes for disease. These are genes that create proteins. And what the disease name is on the previous slide is what happens if that gene is messed up and the protein isn't there. So although we name genes after the disease that happens when the gene isn't working, it doesn't mean that the gene is there to give you the disease. The gene is there to do something essential. And if for some reason it gets messed up, then it manifests itself as a disease, which is kind of a strange way to think about it. But it's just how we have studied genetics in the past. So we're going to focus on three, what we would call sex-linked diseases. This one here, Duchenne muscular dystrophy, hemophilia, and red-green color blindness. So there's a lot of other ones on here, but those are the ones we're going to kind of focus on. So if we think about that, muscular dystrophy, hemophilia, and color blindness. So the muscular dystrophy gene is actually, in real life, we all have it. Everybody has the muscular dystrophy gene. Hopefully, I'm sure, we all have it functioning correctly. And so that gene for us helps our cells make proteins that go in our muscles that allow our muscles to contract properly. If it's messed up, then your muscles don't contract properly. What happens in muscular dystrophy is the protein fibers in the muscles are supposed to slide, and then for contraction and expansion. And in certain forms of muscular dystrophy, they don't slide properly, and then they kind of rip and tear, and they build up scar tissue over time. And the muscles just get weaker and weaker and less able to function. It's kind of a chronic degenerative condition. Similarly, hemophilia. The gene that causes hemophilia, again, we all have, but we have it correctly functioning. And for us, it makes a protein that folds up. It goes in our bloodstream and helps our blood to clot. If you don't have that protein, or if it's not folding correctly, so it's not making your blood clot, then if you have an injury, you can bleed out because your blood doesn't clot. That's hemophilia. Or another one, very actually common in this case, and there may be someone in this room, we're not in a room anymore. There's maybe someone in the class that carries this or has this, this mutation, that we all have the gene for color blindness, but most of us probably have it functioning correctly. The sequence is correct, and we make the protein so that we can put the protein in the retina of our eyeball and detect colors. But if you don't make that protein correctly, then you are unable to detect colors in the retina of your eyeball. So that's sort of a quick rundown about how genes are named and why they're named after diseases when they really don't cause the disease. It's just the result of what happens when the protein isn't working right. All right, so Duchenne muscular dystrophy, hemophilia, color blindness. Those are three sex-linked diseases. Oh, also, sex-linked just means that the disease is caused by a gene that is malfunctioning, and that gene is on the X, or sometimes the Y, but usually the X chromosome. It has nothing to do with sexually transmitted diseases, nothing to do with the reproductive system, or something to do with an abnormality of reproductive organs, nothing like that. It just has to do with the fact that the gene that results in this condition is on the X or occasionally the Y chromosome. All right, so let's move on to this slide here. And we're going to talk first of all about hemophilia. All right, so I've got, I'm looking at chromosomes here from 5 different people. On the left side, we have 3 different females. Okay, female 1, female 2, female 3. And on this side, I have 2 males, male 1, male 2. So these are males, these are females. Right over there. Okay, so how do I know these are males and females? Well, I can tell from the length of the chromosome, there's 2 X's here, right? 2 X's, 2 X's, 2 X's. So these are definitely females because there's 2 X's. Over here, there is an X and a Y because this one is much shorter. So these are boys on the right side. Okay, and it's a very significant thing is that the Y chromosome is much smaller than the X, and explains why these sex-linked conditions are often suffered by males more than females. So let's talk about why that is. So first of all, this hemophilia, we're going to say this H, we're going to use the letter H, and an uppercase H says that the gene is working well and you can make the protein that causes your blood to clot. So make protein to clot blood. Okay, that's uppercase H. Lowercase h is that you cannot make the protein for blood clotting, and therefore you bleed. If you cut yourself, you bleed. You don't clot. You don't stop bleeding. All right. So uppercase H means you're making the protein. All is good. The gene is correct. The sequence is right. There's no mutation. Lowercase h means that there's a mutation. The protein is not functioning correctly when it's made, and doesn't help the blood to clot. All right. All right. So this female number 1 on the left. Here is how I would write her genotype. We're going to write an X to show the first chromosome and an X to show the second chromosome. So that's that one there, and that's that one there. And then I'm going to draw an uppercase H up here. Ooh, I'll control my pen. There we go. To show that this is a female and she has an uppercase H, the correct version of the gene, on both her X chromosomes. So she's making tons of the clotting protein. She's going to be fine. She can clot her blood. All right, female number 2, we would write like this. We write an X for the female and for the 2 X chromosomes, and she has an uppercase H on the first one, and she has a lowercase h on the second one. So I'm showing what version of the gene she has right here, and I'm showing the fact that it's on the X chromosome. This female would be also fine. She would not have the disease because look, she can make the protein, so she can clot her blood. But she would be considered a carrier because she has the messed up allele where you can't make the clotting protein, but she's just not suffering from the disease. All right, and what I'm writing out here are genotypes. I'm showing what alleles these people have for the disease. So this is normal, not sufferer. This is normal too. She's not going to suffer, but she is a carrier. She might not know if she's a carrier, like, you know, for all her life, she might not know. But she is a carrier genetically if we could see her chromosomes. And then right at the very end, very rare, we have 2 X's and a little h on both. And this person would be a female hemophiliac. So a female who couldn't clot their blood. Now, back in probably, I don't know, 20, maybe 30, 40 years ago, this person would die pretty young. Nowadays, hemophiliacs can be treated quite successfully because we can inject the extra clotting protein into their bloodstream to provide them ability to clot their blood. But that's a pretty recent therapy. All right, so that's how you'd write the genotypes for these females. Normally, when you write genotypes, you know, you just write the letters, right? HH, like a homozygous dominant or a heterozygous, you know. But when you're dealing with a sex-linked condition like hemophilia or muscular dystrophy or colorblindness, it's always a good idea to show the X and the Y that the gene is carried on. If the gene is on one of the autosomes, chromosome 1 through 22, you don't have to do that. But on the sex-linked disorders, you should. All right, so then let's look at the males here. So male number 1, he's got an X and he's got a Y. All right, now the X here, he's got a normal allele, uppercase H, so he can make the clotting protein. This is going to be a normal male. This is the genotype of a normal male, right? He can clot his blood. Now, why didn't I write anything on the Y? Well, like I told you before, the Y is basically much, 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 much smaller than the X. It used to be an X, we think, but over evolution, it's gotten whittled away and whittled away, so there's only a little bit of it left. And a lot of the DNA is just missing. It's been deleted from the chromosome. And so where there should be another copy of the H right here, there's nothing to put it on. It's just literally missing, right? So males only have one allele of this gene. They don't have, so, you know, this female over here, big H, she's got 2 alleles, right? And if one's messed up, the other one can make up for it. That's the carrier. Right? But over here, there's nothing to make up for it because there is no where on the Y chromosome to put that gene. It's just gone. It's been erased. All right. So we would just write X, uppercase H, Y, nothing. All right. That's a normal male. Down here, we have X, lowercase h, y, nothing. All right, because this one is here and this one is right here, right? And this would be a male hemophiliac. All right, so up here we have a normal male. Down there we have a male hemophiliac. So the reason why males tend to suffer from these conditions more than females is that they cannot hide them. They can't carry them. If they get the lowercase h allele or the lowercase m allele for muscular dystrophy or the lowercase c allele for color blindness, they're going to have the condition because they have nothing to make up for it on the other chromosome of the pair. The Y can't make up for it. Females, on the other hand, they can carry and they can be totally fine. And it's only in this situation where they're going to actually manifest the disease. All right, so to finish up this part, I'm going to do and because this is getting long, I'm going to show you my family tree because my family tree contains color blindness, and I'll show you how we would work this in a pedigree. Okay, so I'm going to start with my mom, all right, and my dad, and let's just finish off here. And then me and my brother. We'll go with that for now. All right, so this is me. Okay, now I'm a circle. I'm a female here, you can tell, and and I'm going to tell you my brother is red-green colorblind. So I'm going to color him in because that's what you do when someone is a sufferer of a condition. For this, we're going to say my brother is a sufferer. All right, so we know for a start that my brother is male, so we can say X, Y, all right? And let's, over here, I'm going to pick a little key. We're going to say big B is normal vision, and little b is colorblind. All right? So my brother, and this is the color blindness gene is on the X chromosome, so we know it's sex linked. All right, so if we're looking at my brother here, and we know he has normal vision. He's colorblind. Excuse me. So we can say that he must have a little b here. All right, and there's nothing on the Y because it's a sex linked condition. There's nothing to make up for it. If he had a big B here, he would have normal vision. Right? So we know for sure this is my brother. All right. Me, I also have normal vision. So for me to have normal vision, I'm going to draw female. I could be this, or I could be this for now. Both of those people, those conditions would be able to see colors because they've got at least one big B. There's one there, there's one there. All right. Okay, let's go back to the next generation or the previous generation, my dad. My dad could see colors just fine, So we're going to put him here, X, Y. And if he could see colors, we know for sure that he must be a big B, right? Okay, good. Now, let's look at my mom. So I'm going to erase this so that I have some space to write. All right, so my mom, female, so we're going to do X, X. And someone who has normal color vision should be big B, or big B, little b, right? That's same as me. But let's look at a few things. So this is how you have to work these pedigrees. It's kind of like a detective elimination kind of deal. Let's look at my brother. For my brother to be male, he must have got this Y chromosome from my dad, right? He got one of these came from my mom and one of these came from my dad. Well, my mom doesn't even have a Y to give him, because if she had a Y, she wouldn't be my mom, she'd be my dad. So we're going to say we know for a fact that this Y came from my dad, which means this one must have come from my mom, right? So if my mom gave him an X little b, she must have one to put in her egg, which means she cannot be this, because that person would not be able to give a little b to their son, right? Could she be this? No, she cannot be that, because if she was that, she would be colorblind herself, and she's not colorblind. So we can erase that. So we know my mom is a carrier. Now, did she know that? No. Was anybody else in her family colorblind? No, she had no clue till my brother was tested in like first grade and proved to be colorblind. You know, there's little dots you look at and have to look at the numbers, right? So my mom is a carrier. My dad had normal vision. Now let's look at me. First of all, female. So what must my dad have given me? For me to be female, he must have given me this, right? So I know I got at least one big B from my dad. What could have my mom given me? Well, she could have given me the big B, or she could have given me her little b. We don't know. If she gave me the big B, this would be me. If she gave me the little b, this would be me. But either way, I'm going to be able to see colors, which I can. So right now, I don't know. I might be a carrier. I might not. The only way to find out would be for me to have a bunch of kids. And if I had a boy that couldn't see colors, then I knew that I was a carrier. But I'm not going to do that just to find out if I'm a carrier of color blindness. So that's how you would work through a family tree for color blindness. Sometimes you can't resolve it, like with me down here. I don't know what I am, and it's okay to leave it like that, like it's this or that. It's a 50-50 shot. I could be one or the other. You know, we could have gone up here. My mom has a brother who has normal color vision. So my mom's brother must be this, right? And then her parents, her father, my grandfather, also had normal color vision. So this, which means for my mom to be a carrier, she must have got the big B from her father, because if she got the Y from her father, wouldn't be my mom, she'd be my dad. So she must have got the big B from her father. So she must have got the little b from my grandma. So my grandma must also be a carrier. So you can see here how right through my family, there's generations of carriers that had no idea. My mom didn't know she was a carrier. She didn't know her mom was a carrier, didn't know it. It didn't pop out until my brother came along and was colorblind. Now, let's see what happens if my brother gets married, has kids. Hold on. My brother, we're to have kids. Let's say with a female who's a carrier. All right, let's do the Punnett square. This would be my brother's partner. Let's say, for example, if she was a carrier. There's my colorblind brother. It would be big B, little b there. So that'd be a female who's a carrier but could see colors. There's a normal male. There's a colorblind female, quite rare. And there's a colorblind male. So if my brother had kids with a carrier female, there's a 50-50 chance that they would be able to see colors. These ones could and these ones couldn't. And the girls could potentially be colorblind, which is quite not very common, doesn't happen very often. All right, I hope that helped to show you how you backtrack through a family tree, how you use a Punnett square and various different sort of detective information to figure out genotypes in a situation like this.